Okay. Um, yeah.
Like, logistic, like putting inputting the actual grade, not grading it, but just inputting the grade.
I think the TA, like the overall grade or the grades for the homeworks and stuff.
Overall grade. So it's like right now, like one, it's like out of 0.
Yeah.
Which means we basically have extra credit.
Yeah.
It's just not accurate.
Yeah, I think that one we can. Yeah, I can figure that one out by myself. Yeah. Okay. Yeah. I'll. Do you know what the homework one was out of?
I think it's out of 50, but we can just put that.
Yeah. Okay. I'll just make a note to uh update.
I didn't get a perfect score, but you guys got points from it.
Yeah. Yeah. What did you think about the grading? Like, is it fine?
No, one when I probably should talk to the other TA. Nicole graded it.
Yeah.
She said like, how does alternative splicing affect Alzheimer's disease? Which that's the example I use, but like, that's just for homework one.
Mhm.
But I thought that I did use, like that I thought, but maybe I didn't describe it enough.
Maybe, yeah. That's what it seems like from.
Cause like I know I mentioned it.
Yeah.
I thought like I think I like defined what alternative splicing is. Let me read it.
Yeah, yeah. And what was the question? The question is list how alternative splicing can affect like lead to some kind of phenotype or something. Is that what the question is?
What's the question?
I could look up the question too. Okay.
3B. Yeah, so provide an example of how alternative splicing is implicated in a specific disease.
Huh.
So this is just my whole answer. I said it allows us, or alternative splicing allows us to view issues in human neurodegenerative diseases such as Alzheimer's disease. As alternative splicing is common in neuronal cells to maintain homeostasis, there's an increased probability of splicing defects, which lead to the progressive loss of neurons, which results in impaired cognitive function.
Hmm. Wait, can I? So you kind of like from my takeaway from what you wrote, or I guess what it kind of tells me is you kind of you're saying like
Alternative splicing, when it occurs frequently in the cells, that's how we get the when splicing occurs a lot, some of them is just not going to work. So that's the defects.
Yeah.
And those defects will get like there'll be more and more defects as Alzheimer's disease progresses. That was my understanding.
So let me So alternative splicing is not some random event that's happening. It's not like when we look at the gene on the DNA level where you have the boxes of exons and the introns in between, it's not like the cell is just making every single permutation, every combination of the exons. It's very it's very regulated. It's like there are with that one gene there are proteins that are made from exons 1, 2, 3, and that leads to a very specific protein. And then there's ones that are like 1, 2, 4, and that leads to another specific protein. And the way that we describe them in papers is like, let's say it's just like protein ABC1, isoform 1, protein ABC1 isoform 2. It's very specific, and so it's like, I think what you fail to address is that it's not just this random alternative splicing event that's happening in the brain that leads to eventually neurodegeneration or Alzheimer's or something like that. It's actually like, it's the accumulation, or when you look at the population of the different isoforms, it's because the population so let's say isoform 2 is the specific protein that leads to Alzheimer's. It's because there is a greater abundance of that as opposed versus the wild type. Yeah. So it's like, I think that's, I don't, yeah, if you got point taken off, that's why. There's like, yeah, there's a bunch of, okay, yeah, it's fine. It's fine. There's a lot of alternative splicing related diseases where it's like, maybe it's because of, exon 3 instead of exon 4 is in the protein. Maybe it's because it's missing an entire exon completely, or maybe it's because it's an insertion of an exon that's not found in the wild type, that leads to either a dysfunctional protein, usually it's dysfunctional, or it's promiscuous, as in like it's doing something else that we don't want. So it alters the function of the protein downstream. That's what alternate splicing does. Yeah, that's how it causes the problem. Yeah.
Okay. That
Yeah.
makes sense.
Yeah.
She used 2.1 or 1.5, 2.5, which isn't a
Something
lot.
like that, yeah.
I know for me, I think the grading's been fair. I don't know about like, I haven't really talked to my friends about it. I know one of my friends got like a lot of points taken off.
Yeah.
But
It's honestly, grading, maybe you'll experience this one day, but it's so like, while you're grading, okay, like I'm grading the midterms right now, and it's like, I found that I was being super strict on my grading in the beginning, and over time I get more and more lenient. And so there's like this so it's like, it's well, I mean, I feel like people that are being graded in the middle are going to get a more lenient grade. People that have last names with A's, I feel like they're gonna get graded on a lot harder. I don't know. I feel like, yeah. Yeah. And then maybe we can even look at the distribution of scientists and their last names, and then see what that looks like. Because if there's a correlation between, yeah.
Grades, like, I think everyone is like, I would want to do it not for like, just because I'm curious, not because of like any other metric, right? But a lot of like some Asian people have like last names with like X and Z.
Yeah.
So like, I know that they're actually smart and very capable. But I'm also
If it's influenced by that, maybe, yeah, maybe.
It's not actually. I wouldn't use it for anything.
Yeah.
Realistic.
Yeah. No, I mean, it could totally become like some kind of like social research, like, yes, like social, it could be some kind of social science research for sure. No, that, yeah. I mean, I think what will help remove the bias from that is like, once we start getting our like natural language processing algorithms to be able to actually very accurately, more accurately label letters in our homework assignments, then we can just apply the algorithm to be like, and it has to be widely adopted in academia, but at that point we can just apply the algorithm to the homework and be like, find these keywords in each of the answers and assign the grade appropriately.
For sure, I think it'd be a very long time for that to happen, because also I honestly don't like the idea of the keywords.
Yeah.
Because like, I mean, sometimes it's fine, but sometimes like you need it. But like in some classes, I'm like, and then classes that are like physiology or like the medical fields that are like more memorization based, I can see that.
Yeah.
But things where if you spell it or which are just a simple error, but things if
Open-ended questions, yeah.
I mean, it's up to the grader, but like if you're dancing around the topic, if you dance around it good enough, like I wouldn't want them to get no points, even though they just couldn't remember what that word is.
Yeah.
But
Yeah.
I'm sure you're also thinking in the long term.
I mean, yeah,
Very, very long.
right, exactly. So then actually the point you're making is that actually the way that we classify the questions and the answers to our questions, there's a division there. There's there's a different way to cluster or classify the questions and the outputs. And so like, yeah, the algorithm that I'm talking about would be for things that are more memorization based. Yeah. But then you have another, the subjective would need the human touch. Yeah.
So things like seeing like things like ChatGPT, like those ones that actually create the information. How well can they take that information and understand like, oh, if a student's give like a long answer, how correct is that?
Yeah.
So I think I could see that one being more possible. But then, as long as it's accurate.
Yeah. Yeah. I
Then
mean,
it's
yeah.
less looking at the keywords and more. It's like now I think they said like kind of AI, the specific type of AI is like have the intelligence of like a 9-year-old.
Yeah.
Just like really amazing.
Yeah. Yeah. Yeah.
It's absolutely good. So maybe that, but yeah, for sure I do agree that things like A, B, C, or D or
I think with ChatGPT, it's not necessarily, I mean, more specifically, it's not just the intelligence, but it's like the reasoning of a 9-year-old, rather than just the general intelligence. Yeah. I would say it's the knowledge that it has is of, there's not even an age. It's literally the accumulation of all human knowledge, which is, it's beyond uh mortality, maybe. I don't know, unless we can implant the
say human mortality, because that would be
Yeah.
about 100
years. Yeah.
and it definitely has more than. I don't think GPT has access yet to all of that, but very soon.
Yeah. Okay, maybe we should get started on the homework a little bit. All right.
I didn't attend yesterday's lecture. I haven't seen it before.
Okay. All right. So okay, I'm gonna read it out. You want to express erythropoietin EPO in mass quantities. EPO is a glycoprotein. Do you remember what glycoprotein is?
So like glycolysis.
Yeah.
So does it have to do with making energy?
No. So glyco is from glycolysis, but glyco means sugar. So glycoprotein is like is a sugar protein. It's a protein with sugar, we call them trees or branches on them. That's what a glycoprotein is. That's very crucial to this problem. So EPO is a glycoprotein hormone. Hormone is just like, I don't know a lot about hormones, but I think it's just like it's a smaller protein. Yeah. I think it's amino acid based, and I think hormone is actually, it has amino acids and lipids. Yeah. So it's like, but it's responsible for the control of red blood cell synthesis in bone marrow. Okay. So it has invaluable medical usage in anemia and even cancer therapies. The molecular weight of EPO is about 34 kilodaltons and includes highly complex glycosylation. So that's what we're talking about, the sugar trees on the protein, the glycosylation. So the first question is, what type of expression system would appropriately and efficiently express your gene of interest? You have no idea? Just anything, just say anything, even if it's low confidence. What do you think? What's the first thing?
Okay, so I see. So I want to see my gene of interest. Sugar. Is it when like expression system? Is that something like a fungi or like so?
Yeah, expression system. Yeah. I mean, yeah, it is a little bit ambiguous. The expression system can span anywhere from the exactly the plasmid, the DNA, right? The way that you construct your where you insert your gene of interest in what form of DNA, to all the way to the organism that's gonna produce the the end product, the protein for you. So it does span that. So yeah, I mean, since it's a little bit ambiguous, I would probably try to touch upon both of those. I'd talk about, yeah, I mean, I would talk about, yeah. So it's like, okay, so the main takeaway from this class, in terms of glycosylation and expression systems, it looks like those are the two main terms, glycosylation and expression systems, is that there is a hierarchy of post-translational modifications, depending on the complexity of the cell. So since bacteria is the simplest form, the amount of glycosylations or post-translational modifications is the lowest. Is the lowest, yeah. So when she says something like, oh, it includes a high complexity of glycosylations.
Bacteria. So if it's high complex of glycosylations, would a bacteria be able to handle that?
Exactly. That's
It would be too complex and the bacteria wouldn't be able to do it effectively.
exactly.
So it wouldn't really be, it's useless.
Exactly.
So it's easy, but that's the flaw or one drawback of using the bacteria.
Exactly. Yeah. So yeah, exactly. Yeah. It seems like you have a good understanding of that. Yeah. Yeah. Okay. So I think that's what she would want. I mean, in terms of the the genetic vector, the plasmid or whatnot. I mean, yeah, you can talk about that. Let's see.
Would it be good to also, so if I talk about like a plasmid, so basically two parts of this, I would talk about like which way to like a plasmid of some sort and then what organism.
Yeah.
So for the plasmid of some type, should I also include like components that I would include?
Yeah, I would. Yeah, I mean, I think that might be good practice, but when I look at this question more, it doesn't seem like talking. I don't know. I don't know. Maybe mention the plasmid thing, but I don't feel like she's actually in instigating specific details about the plasmid here. It just seems like she's trying to make a connection between glycosylation and um the expression system, the organism, the order of the organism. So I think that's what that is, yeah. But yeah, um your system from the previous question is implemented and the gene is inserted. However, you're not able to obtain the desired protein of interest. What are some possible reasons, and how might you adjust them? Yeah, do you have any thoughts there?
Questions. I mean, it's
Pretty straightforward. Yeah, yeah. Yeah, I think I guess the way that I would tackle this question, and there's many, this one is super open-ended, right? It could be like maybe protein folding is off, maybe it's the expression of the promoter is not good, so we need to increase the expression. Maybe yeah, secretion, maybe there's all these different things, but I think that the one thing that I'd probably hedge my bets if I don't have the answer key, so I don't know what she's expecting, but because the first part is dealing with glycosylation, I would probably talk about how I would obtain metrics on the level of glycosylation in the cell. So I guess how I would address this question is like I would maybe insert genes or something like that that are responsible for glycosylating proteins in the cell, right? To make sure that, if I know that mannose is 90% of the sugar of the glycosylation on this protein, then I'm gonna instigate the mannose production in the cell or all the pathways that are involved with that. And if I find that it's not sufficient enough for my glycosylation, especially if the protein is being overexpressed, then I'll add a gene, and I'll add another gene, or I'll change the promoter of the mannose related genes to increase the production of that, so that, yeah, something like that. List types of RNA therapeutics and their delivery systems. This one's easy, right?
I feel like that one, I could just look at the notes.
Yeah. Okay. Yeah.
Delivery, I mean, if I can't find it in my notes, then I'll just Google the like the delivery systems, but I know some of
I mean, there's one. Do you have one that comes to mind immediately?
No, but I really think I just gonna look at the notes.
What? What about?
It's not really applied right now, but. Well, it's not actually like used medically.
Okay. Yeah.
So, that one's something. Another one is the mRNA, the thing that caused the COVID-19 vaccine.
Yeah. Exactly. Exactly that. I mean, the CRISPR one. Yes. I would think that that is at least.
I feel like I shouldn't use that one just because it's not being used yet as a therapy.
I mean, yeah, I think the thing about the thing about CRISPR is that it's not solely an RNA therapeutic. You have to deliver also the CRISPR enzyme, right? But there are therapies where it's literally you're just delivering the RNA alone, and it utilizes the endogenous proteins uh to obtain its therapeutic qualities. Yeah, there was a lot. I think there was an entire. I think Kun Zhang's lecture goes over that, and there's a lot of different ones. Yeah. Okay. Yeah. So please list the clinical applications of RNA therapeutics again. Yeah. Yeah. I think in Kun Zhang's slides, there's like a timeline of different applications there. What is the first causative gene identified for ALS? Okay, this is also in Kun Zhang's slides. I think there was a slide. Do you remember? Actually, ALS was named after. I think it was like it's like I forgot what it's like. I want.
What it is like physically.
It's just it's muscle atrophy. It's when your muscle atrophies. Yeah, so you lose, or maybe it's not muscle. It might be muscle cells, or it might be the nerves that are connected to your muscles. But you start to your yeah, like Stephen Hawking, do you remember? Yeah, he's an example of ASL. What are strategies for optimizing mRNA vaccines? Yeah, I think you can find that in Kun Zhang's slides as well. Yeah, but if you have trouble with that, maybe we can address it. I mean, I guess I could briefly address this. It's just like, yeah, if you look into his slides, there are things that you can attach to mRNA so that it is protected. Yeah, yeah, that kind of stuff. Yeah. I think also another one is just kind of like the vector that it's used, the vector they use to deliver could also be another thing you can talk about. Oh, okay, never mind. So the next one is list 3 delivery strategies. So I guess you would talk about it there. Yeah.
So would the capping be a delivery strategy because you're protecting it?
No.
Or no?
No.
Yeah. You're just protecting it. It's not really helping with the actual delivery.
Right. Yeah. Yeah. I mean, even if you cap it, I mean, it's what I imagine. Yeah.
Maybe tapping it, and then.
Yeah. Exactly. Yeah. Because I mean, okay, like what I'm thinking is, let's say you have an mRNA, you just have the mRNA for the spike protein, right? And then let's say you have the mRNA for the spike protein, but with the protective cap. If you just simply inject both of those two into your arm, let's say that the uptake into an actual cell of that mRNA without a cap is like 0.001%. And let's say that with the cap, it's like 0.01%. Maybe it's 10 times greater, but I mean, if you compare the delivery of the delivery uh advantages of just a cap versus putting it in some kind of like, I think we talked about a couple of different delivery systems.
But this is kind of like with the protein you put in a capsule.
Yeah.
But
Yeah.
something for it.
Exactly. If you put in some kind of capsule, right, then it increases the delivery system from like 0.01 to like 90%, right? So yeah. So there's that. Okay. So besides fluorescent imaging technologies, what are there any other multi-molecular imaging technologies for mapping biomolecules in situ? If so, what are they? What questions are there? Yeah, I think I think she yeah, I think she wants to, yeah, I think so. I think so. Besides fluorescent imaging technologies, are there? Hmm? Yeah. Yeah, there are other examples. In situ, man, I wish
Like the person or organism.
yeah. I don't know. I'm gonna look it up.
Maybe that's
In its original place. So I guess yeah, in its original place. Okay, wait, let me see. What is the difference? Hmm.
yeah, that's what I was thinking that the just like if you have it like on your
Yeah.
like
So I think the difference would be like in situ is like histological slide. Like you take a biopsy from a person and then you fix it and then you stain and do all sorts of stuff to it. But whereas in vivo is like you look at, I mean, there's a lot of debate over what is the difference between in vivo and in vitro, but I would say in vivo is like for example, Intagliata. I was in his lab briefly, Marcos Intagliata, and what he did was he created these. It's called like TACs, like transparent axis chambers on the backs of like Chinese ovarian hamsters to study the microfluidics of so it's to study the flow of blood through the hamster. So what he would do is you have a hamster, you shave off the hair, and then you kind of barbarically screw on these plates into the dorsal fold of the hamster. And by stretching out the skin like that, you can actually, with the microscope, with the high resolution microscope, you can actually see blood cells moving through the veins. Yeah. So I think that would be an example of something like an in vivo experiment. Yeah. Yeah, where it's still alive. You're the thing that you're the thing that you're observing is still alive. Yeah. And I guess the thing that is a little bit ambiguous is like, what about human cells, immortalized cells on a petri dish? Is that in vivo or is that in vitro? Yeah, it's alive, but it's in like a laboratory setting. Yeah. So, so yeah, some people argue that it is in vivo. Some people argue that it's in vitro. Yeah, I think let's see what this says. Within a living organism. Oh, okay. In vivo is used. outside of a living organism. Yeah, I mean, but the cell is alive. It's just not in a. I guess it's not in technically in an organism. Hmm. Yeah, so maybe growing cells in the petri dish is in vitro. I don't know. That's up for debate. What does bioorthogonal mean? Give three different small molecules that can be utilized using bioorthogonal chemical imaging. Hmm. I think it's yeah, either Thursday this past Thursday or this Tuesday. I don't remember. Honestly, I don't remember this one, but I think you can find it in the notes. Um I think maybe you found it? Wait, non-linear what?
Bio orthogonal nonlinear vibrational imaging.
Wait, repeat that again.
Bio orthogonal
Uh-huh.
nonlinear vibrational imaging. It's not like in the slides.
Mhm.
It's like it's just on the bottom of the slide.
Maybe just yeah, like Google that term or something. I don't know. Let me maybe I
It's labeled grade. It's just alpine.
oh maybe that's what it is.
I think this is Thursday lecture. So yeah, this is Thursday lecture. That's why I don't know.
Bioorthogonal. Oh, okay. I think another word for bioorthogonal chemical imaging is click chemistry. I think.
Without lines is that I don't. I never really thought it was wrong.
Click chemistry. I think if it's the same. Yeah, also term click chemistry is like click chemistry is pretty cool and useful and it's like yeah, I think it's a little underutilized right now. I'm glad it got the Nobel Prize, but it's like yeah, it's like for example, the way that I used it was like so what click chemistry allows you to how I used it was I was able to ligate um um like a DNA barcode onto an antibody, right? And then so let's say that that antibody is its antigen is like an RNA binding protein, right? So that RNA binding protein is going to be regulating a piece of RNA, right? And so then there's a protein, there's the RNA, there's the antibody, and then the DNA barcode. If I introduce a ligase to that mixture, then the ligase will actually stitch together my barcode with the RNA that's being processed by that RNA binding protein. Yeah. And yeah, so that's what click chemistry allows us to do. It's a way of kind of taking two biomolecules and creating a covalent bond between them. Yeah, and so then like when we talk about just proteins, we can create fusion proteins and have them being expressed inside the cell, but like you'll actually maybe I'm wrong about this, but as far as I know, I don't know if the cell ever makes a mixture between can make a mixture between um a protein and a piece of DNA, like one right after the other. But maybe I'm wrong. I might be wrong about that. Yeah. But that's what click chemistry allows us to do. Yeah. Yeah, and I think that one it should be in her most recent slides. Okay. If you're planning to quantitatively image how much percent of newly synthesized DNA molecules derived from deuterium labeled glucose molecules from a snapshot of a single dividing cell. What? If you're planning to quantitatively image how much percent of newly synthesized DNA molecules derived from deuterium labeled glucose molecules from a snapshot of a single dividing cell. I think that might be worded weird. What can be the possible experimental design? So you're trying to okay, so the question basically is like how do you capture newly synthesized DNA using deuterium labeled glucose? And what is the percentage, what is the if you're planning to quantum, what is the percentage of these glucose molecules that you need in order to capture that? Yeah. Yeah. So how much deuterium labeled glucose molecules is needed to capture the the the, the quantity of quantitatively capture newly synthesized DNA, and what would be a experimental design?
It says engine protein synthesis with deuterated amino acids.
Hmm. Yeah.
There's
Hmm. Yeah, what is a possible design experiment? I mean, I don't know. Maybe she does go over something in the slides that like a specific, maybe there's a specific strategy for doing this. And if there is, then just disregard what I am about to say. But I mean, if we know the concentration of glucose, the deuterium labeled glucose that we're adding to our sample, which has our cells, then we can just simply create some kind of serial dilution. So let's say that in one sample we have a concentration of like 1 picogram of deuterium labeled glucose, 1 picogram per microliter of deuterium labeled glucose in solution. And then in the next one I would jump magnitude. So I'd do 1 picogram, 10 picograms, 100 picograms, 1 nanogram, 10, 100 nanograms, 1 milli, 1 micro, 10 micro, 100 micro. I mean, micrograms per microliter. I would kind of span magnitudes like that, and the amount of the concentrations that I gave is way too many. Like you could definitely reduce that, but I think that would be my strategy. Yeah. And then what I would see is since the deuterium can be uh picked up by the Raman microscopy, at what point do I lose signal? At what point, at what concentration am I not able to detect um the newly synthesized DNA? I think that would be my approach, something like that. But maybe she goes over something in the slides that's like, no, actually, if we've worked on this for a couple of years, like a decade or a couple of decades now, and we just do it this way. Maybe there could be something like that. Yeah.
newer stuff. And then there's the thing that she's spent a lot of money on.
Yeah.
One of the variants specific thing.
Yeah. Okay. What do you when you want to quantitatively visualize how much percent of protein molecules are synthesized in a particular liver cell from a piece of liver tissue, what would you do to realize that? Okay. Okay. Yeah. Hmm. Hmm. Do you have any thoughts?
I have a question. For lipids and proteins,
Lipids are the the bilipid layer of the cell membrane. Yeah, so it's like you have these hydrophilic heads, which look more like an amino acid. I would say there's like carbons and oxygens and nitrogens and hydrogens and phosphates and those types of biomolecules. But then in the hydrophobic tail, you just have uh you just have um uh carbon hydrogen you just have carbon hydrogens uh carbon and hydrogen, uh yeah, and some of them are going to be single bonds and some of them are going to be double bonds, but those are lipids.
maybe what she's looking for is like what components you use.
Mm.
So, I think she talked about a lot of yours is sort of like components, but from her exercise, like with a protein and lipid metabolism,
Mhm.
it's label free before protein synthesis. We need the isotope labels.
Mhm.
And we would use carbon source probes.
Mhm. Carbon source probes? What is that?
Maybe, maybe those are like the DAA, so deuterium
Uh-huh.
amino acid,
Uh-huh.
deuterium acetate, D7 glucose, so that's probably one of the specific ones. EPA, OA.
Yeah.
The opposite of that is the non-carbon source probes. So E2O, T2O, some other studies like that.
Yeah.
Those are more like DNA synthesis, protein biosynthesis.
Yeah. Okay. I think, yeah, the thing that is a little confusing to me about this question is, yeah, it's asking for synthesized proteins in a liver cell from a piece of liver tissue. So I'm wondering,
So from, so like liver tissue, would that be in situ?
exactly, exactly, exactly. So this is asking about this is asking about how to identify newly synthesized proteins in situ. Um, I don't, I, yeah, I, I'm, sh- I, maybe she went over this in the slides. I don't remember how. I don't remember how, but I know that, yeah, what you mentioned earlier is a way of uh tracking newly synthesized proteins by using like deuterium or like a carbon-based isotope. But then that means that we would have to deliver that the isotopes to the person while they're still alive, right? Or at least we'd have to inject it. We'd have to locally inject it into the liver uh while the patient's still alive. And then there's the concern about the toxicity of deuterium or those other uh carbon-based isotopes. So I don't know. Yeah, maybe it's in the slides. This one, maybe it's in the slide. I don't know how I don't actually.
Directly related, there's a point that says combining with bioorthogonal probing,
Uh,
SRS microscopy to image in vivo metabolic, metabolic activities in various biological systems.
mhm,
It's like slightly different, because I don't think we will.
mhm.
I think it's probably to do with like bioorthogonal probing.
I mean, okay, yeah. I'm still confused though. I don't know how I would differentiate. Like, okay, let's say that, okay, I mean, my understanding is that with click chemistry or bioorthogonal chemistry, you might be able to um ligate your a molecule that's gonna in a molecule that's going to give you a specific color in the Raman microscopy, but I'm failing to realize what's gonna differentiate a newly synthesized protein from a old protein. Maybe it has to do with like newly synthesized proteins don't have the glycolysis on. I don't know. This one's hard for me. I don't know, but I'm sure it's in the notes.
It's definitely lecture 17, which is lecture 17 books, but that one she split into Tuesday and Thursday.
Okay. Yeah.
That's other one's pretty straightforward.
Yeah, that was yeah. I feel like this homework's kind of easy. It's just all in the less thinking and okay. All right. All right. Yeah. Good. Yeah. Hey, Bryce, can you hear me?
Yeah.
You have any questions about the homework?
Yeah.
For them?
For 1B, I'm kind of confused.
Okay. Yeah, I mean, do you have anything that you're specifically confused about? Yeah.
Um,
Or you want to just go over it together?
yeah, like, I guess I'm just not sure
Yeah.
on, like, all the possible reasons for why.
Yeah. Okay.
Like,
Okay. So yeah, let's go over this together. So you want to express erythropoietin, EPO in mass quantities. EPO is a glycoprotein. So do you know what a glycoprotein is?
it has, like, I don't know, like, links of oligosaccharides in it, you know, the
Wait,
protein, I don't know.
repeat that again. Repeat that again.
Does it have, like, links of sugars connected to protein? I don't know anything
Yeah,
more
yeah,
specific.
exactly. Yeah, it's the sugars that are, yeah, the sugars that are connected to the proteins. Exactly. From glycolysis or glucose. Yeah, it's the same. It's that same, yeah, glycoprotein. So yeah, glycoproteins are a combination of, yeah, sugars and proteins. And do you know what hormones are? Like structurally?
Proteins, I mean, I don't know, proteins that like act on a receptor in the body that causes some change
Yeah,
in
exactly.
the cellular
Exactly.
response.
But I think I may be wrong about this, but I believe that hormones also have lipids on them. So it's a combination of amino acids and lipids. Um, but yeah, I mean, I'm going over those two terms because I believe they're important for this question. That's the critical detail that will unlock this question. But anyways, so EPO is a glycoprotein and a glycoprotein hormone responsible for the control of red blood cell synthesis in the bone marrow. Um, It has invaluable medical usages in anemia and even cancer therapies. The molecular weight of EPO is about 34 kilodaltons and includes high complexity of glycosylations. So again, she's pointing out glycosylations here. What is the expression system? What type of expression system would appropriately and efficiently express your gene of interest? So what do you think? What are some thoughts about this question?
I was thinking like that we probably use mammalian cells because they produce a large extracellular matrix since they're connected to all these different cells a lot of times. And the extracellular matrix usually has a lot of glycosylated molecules. So I was thinking that that it probably makes sense to use those.
Exactly,
Um,
exactly.
but I'm not sure like about like do I need to get into like what kind of plasmid or like what I'm using there?
Yeah, I mean, I agree with you. Expression system here is somewhat ambiguous. Like we do use the term expression system when we talk about, yeah, the plasmids that we're designing, all the way up until the organisms that we choose to express our end product, which is the protein of interest, definitely. But and if you want to talk about the plasmids, I think that's a way to really ensure that you're getting full credit for this question. But she didn't give us the answer key for this homework, so I mean, I would hedge my bets on the fact that she double emphasizes glycosylations. I think what she's expecting, the answer that she's expecting to just um is to address the level of how glycosylation the level of glycosylation is somewhat proportional to the complexity of the organism that we choose to express our protein of interest. And just so the main takeaway is just like there is a hierarchy of post-translational modifications, where the simplest organisms, which are bacteria, arguably bacteria, um, have low levels of glycosylation complexity, and the higher the order of the organism. So when we go into plants and insects, and then all the way up to humans, the level of the complexity of glycosylations is greater. And so, yeah, I think that that's what she's expecting us to address in this question.
Okay.
Yeah. And then part B, your system is implemented and the gene is inserted. However, you're not able to obtain the desired protein of interest. What are some possible reasons and how much you address them? This is a super open-ended question. Do you have any thoughts about how you would try to answer this?
Like, I mean, how many reasons do I need to have? That's my first question. Or that's a
I mean,
question.
as much as you're comfortable with giving, I guess.
Yeah.
Yeah, enough to make you enough to make you feel confident that you're comprehensively addressing the question, I guess. I mean, there are a lot, but I mean, given the context of the question, I think that there is definitely one that is most important. Um, and then the other ones might just be, yeah, yeah, not as important. But I mean, the fact that she's talking about glycosylation in this question, um, I would talk about how I would ensure that my protein is correctly being glycosylated.
Uh, I'm not sure what does that, to be honest. Like,
You know,
what kind of protein does the post translational modifications? I
right,
don't
right.
know.
So I mean, yeah, let's if we kind of dissect glycosylation. Glycosylation is just the addition of sugars to proteins, right? So then that makes me think of like, okay, well, if I know the if I know that, like, for example, um 90% of the sugars that are being added to my protein of interest is like mannose or something, then I'm gonna ensure that the concentration of mannose in my media is high. That's like one thing. And then everything and the handling, the regulation of mannose in the cell is all done by proteins, right? There are pathways. So I would say that I would look into the pathways and I identify specific proteins that are involved, key proteins that are involved in mannose metabolism, and I'd make sure that they're all being expressed sufficiently and stuff like that. That's how I would go about addressing this question, I think. Does that make sense?
Yeah, yeah.
Okay, cool. And then two through um 10, I believe, is can be found in so 2 through 6 can be found in Kunzing's lecture slides. And I believe 7 through 10 can be found in the Raman microscopy slides that Professor Shi has been talking about in the past couple lectures. Um, yeah, so if you have questions about those, it should be pretty straightforward, but if you have questions about those, like, email me, or yeah, maybe if I get a lot of people asking about these questions, maybe I can set up another office hours or something.
Okay, awesome. All right,
Yeah.
thank you,
Hopefully that
Sam.
helps. Yeah. Yeah.
Have a good one.
Yeah, you too. Have a good weekend. Okay, and then